The overall goal of this procedure is to make genome-wide single-gene deletions in Streptococcus sanguinis by high-throughput PCR. This is accomplished by first obtaining 3 amplicons by high-throughput PCR amplification, including the antibiotic resistance gene, as well as the upstream and the downstream DNA sequences of the target gene. The second step is to obtain the linear recombinant PCR amplicon using 3 PCR amplicons as templates. Next, the component S. sanguinis cells are prepared. The final step is to transform the linear recombinant PCR amplicon into component S. sanguinis cells. Ultimately, colony PCR amplification and DNA sequencing are used to verify the correct mutants.
The main advantage of this technique over existed other methods, like suicide plasmid knockout, is this method can create the genome-wide deletion mutant directly use high-throughput PCR.
Primers can be designed and synthesized as described in the written protocol, using in-house scripts based on the Streptococcus sanguinis SK36 genome sequence. 3 sets of primers were designed for amplification of the 1 kilobase upstream sequence of the target gene, the APHA3 gene encoding kanamycin resistance protein, and the 1 kilobase downstream sequence of the target gene. Gene-specific primers R1 and F3 are designed based upon the